Our next panelist is Matt Miller from Reneco International Wildlife Consultants. Matt's going to tell you about his work, some of the world's largest conservation projects.
Yeah, hi. Thank you very much for inviting me here to speak. I'm really excited to be here. I work for Reneco International Wildlife Consultants, and we run the largest vertebrate captive breeding program in the world out of UAE. We breed various birds that are part of the traditional Arabian falconry. And so you had birds like the Hubara bustard, which in the 1950s through 1980s suffered a very severe decline and was at risk of going extinct. And through the leadership of the Abu Dhabi government, they've invested quite strongly in a captive breeding program, which has resulted in a lot of basic science, including genomics work. And so I'm going to talk a little bit about how our genomics work has informed that stuff and where we're moving forward with it. So what we recognized is that for us, in order for us to manage birds like the Hubara in their native range, we had to understand their population structure. We had to understand something about their demography. We had to understand how inbred they were, which meant that we needed to do genomics, which meant we needed a reference genome. And typically, reference genomes are these very difficult affairs. Increasingly, over the last few months, it's becoming more and more easy. But in fact, we're thinking that we can do this even easier and faster at scale. So I want to point out that every genome that you've ever looked at is a model. And just like a map, no genome is perfect, right? And so I showed here a map from the 1500s of the New World, and you can see just how wrong it was. But this map was good enough to allow navigators to cross oceans and to successfully explore the New World region. And in the same way, these genomes that are good enough we can use to understand genomics, especially from a biodiversity standpoint, because what we're interested in is large contigs of ungapped sequence so that we can do various demographic and inbreeding analyses. So here's an example of 4 genomes that we've worked on recently, and what I want you to see in there is we have very high BUSCO scores, and we have extremely long ungapped contig N50s, okay? And you'll see there's 2 birds there and 2 fish there, and that's really important, the fish, because this is how we're pivoting. So in fact, you'll see that we had about 30x coverage, and these are very good genomes, and they're not at chromosome. We could make them to chromosome. You can always improve your genome, but these are very good, and we were able to generate these genomes from sequencing to draft genome in a week or less with a team of 2 or 3 people. And these cost, in terms of consumables, at least 1/3 of what a traditional platinum-level genome would cost. So this allows us to do it at scale. And in fact, the thing that I want to point out here is, and this is where that circle chart is there, our genomes, although not technically classified as chromosome, have longer fragments. If you take a fragment of 30 million bases ungapped as our N50, only 6% of bird chromosome-level genomes achieve that metric. And so ours are able to do that in a very cheap way. This scales, and in fact, what we're trying to do is to sequence all of the native fish genomes from the Red Sea. And the only way we can do that at scale and quickly over the next 3 years is by taking this fast approach. So if you have any more questions, see me about how to do this. I'd be very happy to talk about how you guys can also do genomes good enough for conservation.
Thank you, well done.
Thank you, Matt.